Guys, welcome back to my lectures. In today's lecture, we are going to see gene transfer in bacteria. How gene transfer is done in bacteria. Before learning the gene transfer in bacteria, you should know the structure of bacteria or bacterial cell structure you should know. So, bacterial cell structure includes cell wall, cell wall, plasma membrane, capsule, ribosomes, pillars, cytoplasm, flagellum, nucleoid DNA, and plasmid. So these are the basic structures of bacterial cell. Cell wall is the outer covering, plasma membrane lies behind the cell wall, capsule is present for their defense mechanism. Ribosomes are present to synthesize the proteins. Pillars are the locomotory organ. Cytoplasm is present for the locomotion or cytoplasmic streaming. And flagellum is one of the locomotor organ like pillars. And nucleoid DNA, that means there is no specific shape of DNA is present, thus crumbled form of DNA is present in the bacterial cell. And one more important thing present is plasmid, which is nothing but the extra chromosomal DNA, double stranded, which is physically separated from the bacterial DNA and thus named as plasmids. So you can define plasmids as plasmids are nothing but the double stranded extra chromosomal DNA present in the cytoplasm of bacteria, which are responsible for replication purpose, which have the ability to replicate one bacterium to another bacterium. So according to the depending upon the structure and functions, plasmids are classified into 5 different types. First one is the resistant plasmid, second is virulence plasmid, third is degradative plasmid, fourth one is col plasmid, and last fertility F plasmids. The complete video on the structure and functions of plasmid will be uploaded on my YouTube channel. Now, horizontal gene transfer. Bacteria have mechanism of gene transfer in the form of horizontal gene transfer. So what do you mean by horizontal gene transfer? It is nothing but the lateral gene transfer in which an organism transfer genetic material to another organism which is not its offspring. That means the genetic material is transferred laterally in between two different organisms. It helps to cause large scale changes in bacterial genome. And the horizontal gene transfer in bacteria is carried out by 3 different mechanisms, that is transformation, transduction, and conjugation. Basically, most of the time conjugation mechanism is preferred by the bacterium which replicates DNA truly, you can say. Now let's see, in today's lecture we will see only transformation, and transduction and conjugation mechanisms will be discussed later. So transformation is nothing but the horizontal gene transfer method in which one organism transfer its genetic material to another microorganism or bacterium laterally. Okay, so through the diagrammatic representation we will learn the transformation. Before learning this, you should know different important factors needed during the transformation. First one is the competent. Competent is the stage, or you can say competent cell is nothing but the cell which has the ability to transform itself with other microorganism. Okay, then second one is the SSDP, that means single stranded DNA binding protein, which helps to bind the donor's DNA to recipient DNA, and which prevents donor DNA from degrading by the enzymes present in the cytoplasm. Okay, then third one is the nuclease. Nuclease enzyme is used for the digestion of donor DNA. And fourth one is the rake A protein, that means here recombination process will be occurring because we have to join donor DNA and recipient DNA so that new plasmid will be generated na. That's why rec a protein, that means recombination a proteins, helps in the recombination. Now let's see one by one how transformation is takes place. Okay, so in first diagram you can see there is a bacterium. Whenever it has to compete or it has to replicate with another bacterium, it gets die. First bacterium gets die, that means donor DNA, that means you can say donor bacterium gets die. Then uh, after its death, the fragments of DNA from the dead bacterium will be generated. That means after the death of the first donor bacterium, the DNA gets degraded or denaturated. Then coming to the second, these fragments of DNA from dead bacterium will attach to the transposomes present in recipient bacterium cells. Okay, the second one is the recipient. second one जो bacterium होगा वो recipient bacterium होगा जिसके ऊपर transformosomes होंगे जो फर्स्ट बैक्टीरियम के डीएनए को अटैच करने में हेल्प करेंगे. सो जो भी डिग्रेडेड डीएनए फ्रेगमेंट्स होगा फ्रॉम द डोनर बैक्टीरियम विल बी अटैच टू ट्रांसपोसोम. देन competent specific single strand DNA binding protein will help to bind these fragments of DNA to these transposomes. Okay, then what will happen to the next, the one strand of the donor DNA, two strands will be attached to the transposome. Then one strand from the donor DNA will be degraded by nuclease enzyme present in the cytoplasm of recipient bacterium. Then rec a recombinant a protein will helps in joining and attaching and forming new DNA strands in recipient DNA. You can see in the diagram, you can see in the fourth diagram, rec a protein will helps in formation of new recipient DNA strand. Thus new plasmid will be generated containing recipient DNA as well as donor DNA. In this way transformation is occur in bacterium, that means competent bacterium most of the time, which the bacteria which have the ability to transform itself from one bacterium to another bacterium. So this is the process of transformation of horizontal gene transfer. Thanks for watching.